But welcome everyone and congratulations for being able to make it and sustain your attention into the second day, in particular to those who were here for the hackathon or the training. It's been a long week, so congratulations for getting this far. So one of my favorite features of Nextflow is its provision of utility across a wide variety of workloads. I mean, it was very convenient actually that Angel sort of briefly touched on this in the talk just before. We might consider use cases for Nextflow on an axis of regularity. On the left-hand side of the spectrum, you might have researchers and scientists using Nextflow essentially as an executable lab notebook. Features such as caching, containerization, Git integration, and the Wave service to provision containers on demand are all sort of useful in this context. Here on the left-hand side, Nextflow essentially allows you to try many ideas in quick succession and work at velocity, and you know that Nextflow has your back, so you're never sacrificing reproducibility. On the right-hand side, we've got organizations running standardized workflows at massive scale, using Nextflow's portability to run on the cloud or large infrastructure, and perhaps leveraging the Seqera Platform API to trigger and monitor hundreds or even thousands of workflows. Like for this next 20 minutes or so, we're going to talk about Data Studio as a new feature in Seqera Platform, a feature that's probably going to be of most interest to those working in the middle and towards the left-hand side of this spectrum. So Nextflow is a phenomenal batch workload orchestrator, but there will inevitably be occasions where you need a human in the loop to transform or make sense of the data. I'd like to believe that novel science is still out of reach for LLMs, although as a Nextflow developer, I saw the GPT plugin that Paolo demoed yesterday, and makes me a little bit nervous, but I need to check that out. For example, when it comes time to get your human hands a little bit dirty with some interactive analysis, you might want to take, for example, the outputs of a Nextflow run into a Quattro notebook, or to produce a report or conduct an interactive analysis. Alternatively, you might want to take the outputs of one workflow through some manual training, for example, to feed that transformed data as input into a second workflow. Data Studios provides an opportunity to sort of bridge this gap and bring a reproducible, containerized, and interactive compute environment to the data. If your data is already in blob storage, pulling that data onto a local machine can be cumbersome and error-prone. Like we can instead continue the Seqera platform of moving compute to data. We can use the Fusion file system to mount that blob storage data into the running Data Studio and treat it just like a local file system. The public preview of Data Studios includes support for VS Code, Jupyter Notebooks, and RStudio. And I think most people in the audience have a pretty good idea about what these environments are and how they work. And as popular as these environments are, we know that there are many workflows that don't fit quite so neatly into one of these boxes. To support other tools, upcoming versions of Data Studios will bring support for custom containers. And it's that I'm going to show a demo using one of these custom containers. So this is coming probably later this year. It wouldn't be a Nextflow summit, of course, if we didn't do some high-stakes live demo using unreleased beta features. So we're going to run through an example of how you might use one of these custom Data Studios to accomplish a specific research goal. As an aside, these Nextflow summits, I think, often tend to reflect some prevailing trends in research. And at this particular summit, just yesterday, we saw gigapixel image data from Syme, multiplex immunofluorescence data from Brenner, MRI imaging from the irrepressibly festive Sherbrooke team, the MPX imaging from Alvaro, and the PixelGen team. So I think it's fair to say that imaging and spatial omics is white hot at the moment. So in that vein, I'm pulling this paper. It's not one of my papers. It's an excellent preprint released just last month. The authors used a technology from molecular cartography to spatially resolve and quantify transcripts in maize, leaf tissue, to better understand leaf development. So the specific biology of this is not critical. Like I love plant biology, I love plant genetics, but we're going to use the Seqera platform to very roughly reproduce a small section of this paper. I'm essentially Bob Rossing this really excellent piece of scientific work into a 30-minute talk to showcase particular Seqera technology, and I hope that the original authors will forgive me for any sort of generalizations I make about the paper. So here is a small section of the maize leaf tissue directly from pulling data from the paper. We've stained the cellulose, cellular walls here in blue, and DAPI stained the nuclei here, shown here in magenta. The molecular cartography system is an optical platform to spatially resolve single transcripts, which we can overlay onto these two strains. So each of these yellow pixels is an individual transcript, which we have XYZ coordinates, and a transcript ID. I'm just grouping all the transcripts together, but just imagine in yellow, just for convenience, but imagine they're all individually labeled with a transcript ID. So by summing the counts over the whole image, we could determine the mean transcript profile for the slice. But I mean, bulk transcriptomics, what is this, like 2020? We can turn this data into a single cell experiment by determining the boundaries of each of these cells and then grouping the transcripts by cell ID. From here, we can follow a really similar pattern to the one that Brenna showed yesterday. You can cluster the cells by transcriptional profile, maybe overlay those clusters on the original image to understand the spatial relationship between cell types and transcript abundance. But for our purposes today, we're just going to try and focus on this one particular task of finding these cell boundaries, the task of image segmentation. So looking at this image, you might think, oh, that's not so bad. I could probably just about do these by hand. The problem is that this can quickly get overwhelming. So I'm looking for an Nextflow pipeline of the right size to run live. Unfortunately for me, we have NF-core Molcart. So this is one of the really great spatial transcriptomics pipelines that are provided by the NF-core community. And this tube map is a really simplified version of this Molcart pipeline, focusing in particular on the cell pose track. You can see it in the top right there. And we have some automated models for cell segmentation, but they often don't perform that well when the test and the training data are too different, which is like a classic machine learning problem. CellPose has a segmentation algorithm, has a base model that performs relatively okay, but it actually performs spectacularly well if you do a little bit of manual training and provide a small, manually curated training set. So we can take these two layers on the top left, and we're going to pull them apart and feed them as input into our pipeline. These images flow through the pre-processing, then into segmentation with CellPose and mask filter, before joining the transcript spots, where we do cell assignment and post-processing. But you'll notice that the CellPose method there takes this custom model as input. And this model can be produced by training. The pipeline also conveniently has this training subset feature, or sub-workflow. And we're going to use that to subsample the original image to generate this training data. But there exists this gap where we need to inject some human intervention. Like the Molcart pipeline is great. It does almost everything we need, but there's just one little gap that we need to fill. We need to turn that training data into a custom model, and we need real eyes and real hands to do it. So, of course, this is a perfect case for data studios. So that's the demo we're going to show. So step one, generate the training data. Step two, we're going to train the segmentation model. We're going to take the outputs, which have been written by the Molcart pipeline directly to blob storage into a data studio instance. It's going to look like local file systems. It's just like we're going to be running on our local laptop, and we're going to turn that into a custom model. Then step three, we're going to take that model and rerun the Molcart pipeline from go to woe to do the cell segmentation, spot processing, and all of that downstream analysis automatically. Optionally, of course, you could then continue this pattern, take the outputs of that into a data studio for more manual transformation. Maybe you want to do the clustering. We're not going to do that today. We only have 25 minutes or so, so we're certainly not going to do the whole paper. The original authors had a little bit more time than a 20-minute on-stage demo. So I'm just going to quickly switch screens. Actually, that's better. Great. So we have these three stages. Step one, generate training data. Step two, train the model. And step two, apply model to data. It's a shame. So here we have the Seqera Platform, and we have a launchpad that I have set up with the bulk RNA-seq and the Molcart pipelines. So the first thing to do, step one, is to generate the training data. So we're going to launch Molcart. Launching a pipeline in Data Studios is really easy. It's going to drop it down, so I'm going to give this a name. I'm going to call it generate training data. I'm going to use CellPose. That's perfect. That's perfect. Under training subset options, I'm just going to click this. And the input we're going to use I'm going to browse and use the datasets feature. We have a sample sheet that I pre-prepared, which points to some training data that's sitting in S3. We're going to look at that training data in a sec. I'm going to specify the output dir to be, we're going to look through these buckets, and I'm using the Data Explorer feature here that's built into the platform to drill into our buckets. I'm going to select a path, just paste it on here. Great. I have an out dir, and I'm happy with that. And click Launch. So that's going to submit a job to the AWS Batch APIs. It's going to spit up an instance for Nextflow. Nextflow is going to start and submit all of those component jobs. It's going to take a minute for that instance to spin up. So I ran this last night. So this is what the run would look like. So we have a classic Nextflow run, pipelines, and included are these reports. And included in those reports is a multi-QC HTML file. Let's give ourselves a little bit more room here. And this is the original training data. You probably recognize it from the slides earlier. And we have the algorithm that has selected these 4 sections for training. So I'm going to take those 4 images to do some manual training in a data studio and then feed that model back into the pipeline. So let's double check that those outputs have been written. I can use the Data Explorer again to see our bucket. Step 1 outputs. This is my run last night, and I have my 4 training images. Now, I have 2 options here. Either I could download those images locally, I could pull them onto my machine, but then I have the problem of, what if I update the software on my machine? I want to be able to always have this machine as a flexible development environment. Things are changing all the time. But for this work, I want to have a stable environment. So I'm going to use the containerization that is necessary for data studios and pull this into a data studio that already has all the tools I need installed. So here we have the Data Studios pane. You can see some of the data studios that my colleague Florian has set up for us. And we have a custom data studio. This is the one I'm going to use. And I'm going to just simply connect to this data studio. Now this is a custom container running some desktop-like software. It's running Xpra. And I have all the usual bells and whistles. I have a terminal. This is just like a local machine. I have a file explorer. And you'll see, if we inspect the details of this, you can see that I have mounted 2 data links into this data studio. These data links are essentially pointers into blob storage. One, I've mounted the root of a bucket, a private bucket that is located within our account. And secondly, I've also mounted some public data. Here, some 1,000 genomes, a particular directory, a particular sample in the 1,000 Genomes project. And those are reflected in the File Explorer here, just as if they're local data. So here I have our training subset. I can open up these images. I could view them with the image viewer, but actually let's just load up CellPose, which is what I'm going to use to do the manual training. I'm going to load up an image. Under data, we have our bucket, as if it was a local file system. Great. Here I have our image. So the first thing I'm going to do is I'm going to segment around green, do a quick calibration, and then run Cyto3. OK, so this is not performed too badly. You can see here the cell boundaries. So this is the base model algorithm, and it's generated this segmentation. But it's not quite perfect. I can see, for example, here, that there's probably 2 cells instead of 1. So I'm just going to select that, remove it, do a quick little, yep, that'll do. And one around here, that looks much better. OK, so I'm happy with that. The next thing is to start training my custom model. I'm going to train a model. I'm going to call it Summit model. OK, and here we're using the GPU on this instance to do the training. This is much faster than doing it without the GPU. Second, we move on to the second image, and I'm going to conduct the same sort of process. I can see here that this is probably 2 images as well, so this is 2 cells. So I'm going to select this, remove that, and do a quick segmentation across here. I mean, if you've ever done this before, you're probably looking at me and thinking, this person doesn't know what they're doing, and you are absolutely right. I'm a genomics person, and I actually don't know anything about microscopy or spatial imaging. But fortunately for me, we have Florian, who's sitting here in the front on our team. And I'm actually just realized that I'm way out of my depth here, and I would actually like one of my colleagues to step in and try and finish this cell segmentation for me. So rather than having to pass him the data, perhaps resolve IAM permissions, copy the data to him, then make sure that he gets it all, do validation, I'm simply just going to take that URL into Slack and just pass it to him. And now we've set this model up to do with sharing features. So this is actually, he's going to annotate this live on my running Data Studio instance via this URL. So you can see this is now Florian doing the segmentation, not me. So while he's doing that, I want to talk a little bit about sort of what is happening here. It's worth knowing that, firstly, there's no need to shuttle data back and forth. And I think this is the big advantage here. Every time you move data around, it's expensive depending on the data sizes. Of course, we're just dealing with relatively modestly sized TIFF images here. But of course, this is a crowd that knows that this data can quickly get out of control. And the more time you spend moving data around, it's just time wasted and potentially data lost. I'm also love the fact that these are stable environments. Great segmentation work, Florian. They're worthy of an applause. It's really lovely to have a stable environment. This Data Studio is checkpointed, so I can go roll back at any time I stop and restart this Data Studio. It checkpoints the state of the running machine for me. So at any point I can roll back and pick up where I left off. But more importantly, for my experiments, I can have a stable point and just restart the machine for each of my experiments at that same point. Same software, same versions, everything locked in the container. You also can provision the resources you need. For example, this particular algorithm runs spectacularly poorly on this very expensive M3 machine because everything is CUDA. It's about 30 times faster on this relatively modestly sized GPU instance from AWS. So it's lovely to be able to suddenly have a 32-core machine or a 128-core machine or a GPU instance at the drop of a hat. The credentials are also managed for me. I don't need to worry about permissions because I know that Florian has access to my workspace. He can do the work. And lastly, we get that lovely shareable URL just to quickly pass data between members of a team. Looks like Florian's done. Thanks, Florian. So let's just double check that he's done the work. Inside of the compute environment, inside of the Data Explorer, I can go back into our bucket. We have the step feature, and we have the summit model here. So this is the same data that he was writing to what looked like a local disk is reflected on S3 and directly inside the Data Explorer. So great. Oops, now it's time for step 2.2, where we're actually going to apply the model to the data. So I'm going to go back to the launch pad. I'm going to launch Molcart. Oops. I'm going to go back to our Data Explorer and just grab the URL or the path to that model. Just copy that to the clipboard. I'm going to start Molcart. Final step. I'm going to paste that S3 path into the custom model. I can optionally change the diameter of the cell, many other parameters that I want. And then when happy, I can click launch. And that's now pulled the manually trained model, and it's going to pull it through that pipeline. Again, I'm not going to make you sit here and wait for this to finish. So here's one that we did a second ago. It has, of course, the normal multi-QC reports. I can see this is just one sample, so it's a bit brief, but you can see the total number of cells, the average cell area. But, of course, spatial omics, it's much more satisfying to actually see the data. So we get spin back into our Data Studio, the same Data Studio we have running, and I'm going to run Napari. It's a really handy little tool for visualizing these models and the results. I'm going to open up a file. I'm going to go back to our workspace, our data, our Nextflow Summit, Molcart final. You can see the stack. So this is the nucleus and the cell wall staining. Let's just split that and make it a little bit more colorful. And lastly, I'm going to open up the files and look at the results of the pipeline, which is this segmentation from CellPose. Great. So here I can see the results of the model. And again, if you know anything about cell segmentation, you know that this is not perfect data. We could probably do a better job here. The original authors did a fantastic effort of segmenting out the whole slice and training models for each individual layer of this leaf tissue. But obviously, we're not going to do that right now. The point here is to show that this could all be done without leaving the Seqera platform in quick succession to get these results. So those of you who are perhaps a little more genomically inclined, I hope that the value is there and you can understand how this might be transferable to your use case. But of course, this is an any old container, so we can install whatever we want in here. So I could also install, for example, IGV. It's often the case where you've run a pipeline and you have a BAM file or you have some regions and beds and you want to quickly visualize it. Yes, you could download those files, but if you're dealing with large BAM files, that can become quite a pain. So you remember here that I mounted 2 buckets into this data studio. I mounted the bucket that sits in our account, that contains our data private to us, where we conducted that manual transformation and training. But I also mounted the 1,000 genomes bucket, so I have all of that public data, and that will work for any public bucket. So let's load in here in the 1,000 genomes, an alignment. Let's load in this chromosome 11. So very easy. Maybe long non-coding RNA is not the most exciting piece of genetic material, but that works just like it does locally. But more importantly, I can go back. That was how big was that file? That was 700 megs, loading very quickly, but we can just load huge files, this 185 gigabyte file, as if it's local data. And it loads like that in a flash. Of course, a lot of that work is because of BAI files and the indexing, but the Fusion file system is seeking into that file and just pulling the data we need. So if you want to quickly check enormous BAM files and your current workflow is to move that data from blob storage to your laptop, wait for it to download, and then perhaps move it up again, I think this is a much more compelling way to do this. So with that, the last things I want to recap are the benefits here. So we're not moving data back and forth. We're using Fusion to mount the bucket. We have stable environments for this work, for the interactive work, which matches the stable environments you're using for your Nextflow workflows. You can provision the resources you need, from small instances to run quick analysis, all the way up to massive GPUs. The credentials are managed by one person. Perhaps it's your cloud engineer who's done that work. Your bioinformaticians shouldn't be thinking about infrastructure. They shouldn't be thinking about permissions. One person can set up for the workspace, and then everyone can get going with their real work. We have checkpointing of these Data Studio instances to move back, roll back and forth. And you have a shareable environment with a simple URL. With that, I want to thank the Nextflow team, the engineers and Seqera for putting this product together, and in particular to Florian, who's really demoed this and I'm just the person who happens to be presenting it. But if you have any questions about this, feel free to drop by the Seqera desk. Myself, Florian, and the Seqera team would be very happy to answer any questions. Thanks.
Wow, that's a very cool demo and great teamwork, guys. Very well, and everything went well. That's so cool. Okay, so there's some chatter that I have to get through. Do you have a reference to the paper or copy of your slide somewhere in public space?
I will post it immediately to that's a good idea. Yeah. It's a great paper.
Is there a plan to have a web interface of some sort that will let somebody like a pathologist select good, bad images, then to automatically rerun the pipeline only on bad images with a different set of parameters?
Not built in. I think like our approach here, I think, is to give you the tool, err on the side of flexibility rather than predefined environments. Of course, if that's what you want, you could easily build it on top of this, but it's not, I don't think, a feature of the platform, because that's a little bit too niche, and we're aiming for generality here.
That's fair. So long term, would this work with an arbitrary container to allow interactive steps in a pipeline?
That is an interesting question and a complicated question. I think it's probably a question for Paolo.
Okay.
I think at the moment, I think it makes more sense to keep them separate.
Yeah. Lorena comments, it would be great to have IGV web app as an optional data studio server. It is the server side of desktop IGV and works very nicely to share genomic data.
Yeah, it's a great question. IGV is fantastic, and of course it works in the desktop app that I've shown here, but you could also use IGV inside of an RStudio or a Jupyter notebook. If you didn't like the desktop version, that's totally fine. I think the better version for you would probably be the Jupyter notebook.
I've worked a bit in the Jupyter notebook with IGV in the Jupyter notebook. It's really nice for teaching examples. It's really nice for capturing, like as part of analysis, capturing kind of snapshots, interactive snapshots of the analysis, for recording what you're doing. I would say that's a really cool use case.
Yeah.
It is more limiting compared to like full desktop IGV though. If you're actively doing your exploratory analysis, I think it might be a little of a constraint. But yeah, having both options would be fantastic.
Yeah.
Vlad suggests JBrowse as an alternative, and that kind of makes me wonder, like, what's the bring your own app perspective on this? Like, if I have some other, browser app that I would love to.
Yeah, if your app serves on a port, there's an environment variable that you have access to when you construct the container. Just point your app serve on that port, and you're pretty well. That's the idea. Is you can bring your own app, serve it on that port, and the Seqera platform will be listening for your app, and it'll get forwarded through.
Could you set up RShiny dashboards in that kind of environment?
Technically, yes. I don't think we've done it yet, but it's on the roadmap.
That's a popular thing.
Yeah.
Yeah. But would you then you would want a way to like publish and expose that to others?
Yeah. So
Cool.
at the moment, the idea is that you keep it inside of the workspace and you're sharing it with the participants of that workspace. The whole model here is that everyone in that workspace has shared access to those resources. So if that's what you want, yes.
Cool. Very cool. You rock. Great talk.
Thanks.
That's one of the comments. Okay, one final one, and then are all the buckets you're mounting in the data studio in the same region?
No. The 1,000 genomes bucket is located in Europe, and the other buckets are located in US East 1. Of course, if you are moving data across regional boundaries, there are, like, I wouldn't move huge amounts of data. Again, move compute to data. That's like the theme here. But it's totally possible.
Okay. And one final very serious question, does it run Doom?
It actually does run Doom. That was like shortlisted for the demo. In fact,
thank you for making the decision that you made.
We actually have Florian here has booted up a game. So yes, the internet is too bad. So yeah, the Wi-Fi is not perfect here. But yes, it absolutely does run Doom.
Fantastic. All right, thank you so much, Rob and Florian. That was fantastic.
Thanks all.